Good morning, all. You're all welcome to your e-learning today. Let's start. We are quite on time. Okay, let's pray. Father, we thank you for bringing us again to our class this morning. We ask, Lord, that this time will be impactful and you give us quick understanding and retentive memory in Jesus' name. Okay, the last time we were still looking at microorganisms around us. Last week we started the topic. It's still a continuation. We are still continuing the same topic, microorganisms around us. So let's look back at what we have learned before. We defined microorganism as any tiny, small organism that cannot be seen with an ordinary eye, but with the aid of a microscope, isn't it? Then we also looked at the categories, that's groups of microorganisms. We said that microorganisms are categorized into 5. We have bacteria, we have fungi, we have viruses, we have protozoa, and then we have algae. We looked at viruses, bacteria, protozoa, and algae, and then we went further to look at culturing. Pathogen, don't forget, we also said that pathogenic microorganisms are microorganisms that are capable of causing diseases. They are pathogenic, while non-pathogenic are the harmless, but the harmful microorganisms are called pathogenic microorganisms. Then we looked at culturing. What is a culture? A technique of growing microorganisms in the laboratory. We looked at the steps, and then I gave you an assignment. To list out the precautions to be taken during the preparation of a culture solution in the lab. So this morning, quickly, we'll continue from there, and then we'll look at the ways or method of measuring microorganisms, and then we'll also look at beneficial effects or benefits of microorganisms, and then harmful effects of microorganisms also. So let's take 1 or 2 questions. I'll call your name, and then Ashilere, please tell the class, list 2 pathogenic microorganisms you know. Is she there? She's not saying anything. Ashilere. Is it the network? Ashilere, can you tell the class, mention 2 pathogenic microorganisms? Please, if you can hear me, nod your head, because Ashilere is not responding. Kosi Sochuku Umar, can you hear me? Umar?
Yes, ma'am.
Yes, ma'am.
Okay. Ashilere is not talking. I don't know if. Okay, let's just.
Is the network.
Okay, network. Okay, let's start. Let's start the class. Later we can take questions. Now, precautions to be taken during preparation of culture solution. I know most of you you did the assignment and you sent to my mail. That is for few. So many did not. But let's take a few. In case you don't have these ones in your notes, you can as well add them up. Precautions to be taken during preparation of culture solution. Number 1, wash your hands or wash hands with soap before and after the preparation of agar solution. you wash your hands with soap before and after the preparation of what? Agar solution. That is the first precaution you take. Before you prepare a culture solution, you wash your hands before and even after. Now, what else? Number 2, the Petri dish. The Petri dish should be opened slightly and cover at once. The Petri dish should be open slightly and cover at once. That's, you don't leave your Petri dish open so as to prevent unwanted other microorganisms. Don't forget last week we kind of discussed, and I said that microorganisms exist everywhere. You find microorganisms in the air, water, on the surface of your skin, everywhere. So aside from washing your hands, what you do is you cover, make sure that the Petri dishes are always closed. They are always closed until you want to inoculate or introduce the specimen. And after doing that, or in the process of doing that, slightly you open the Petri dish and you also cover it. Avoid talking and coughing or sneezing when preparing it. When preparing it, please avoid talking or coughing or sneezing, because microorganisms exist in our mouth. So if you want to view or you want to grow another microorganism from another specimen, by the time you're talking or coughing, you can contaminate that agar. Now, all instruments should be properly sterilized. You sterilize them, put them inside a sterilizer to kill other microorganisms that must have been on this instrument. So all instruments should be sterilized properly. These are few precautions that we have. Now, let's come back to today's business. Carriers of microorganisms. Carriers of microorganisms. Please and please, I want you to on your video. Joseph Agungu, Williams, Lillian, Jethro, Barnabas, Ulebe, Praise. Please on your video. Put on your video. Kelechi and Israel, please put on your video. I can see Abdulaziz, Jemima, and the rest, but I cannot see the ones I have mentioned. Please make sure your video is on. Now, carriers of microorganisms. Carriers of microorganisms are agents which can transfer microorganisms. They transfer or they carry microorganisms from one place to another. Carriers of microorganisms are agents which can transfer or carry microorganisms from one place to another. Now, these agents include living and non-living agents. Please get that right. You know, we are discussing carriers of microorganisms. I said carrier of microorganisms are agents that pick microorganisms from one place to another. Now, these agents or these carriers, they include living and non-living things. Now, the living are animals. Specifically, they are animals. They are able to carry microorganisms from one place to another. While the non-living agents include air, water, and our food. Air, water, and our food. These are the non-living agents. Now, let's look at the other aspect. Now, I said that carriers include living and non-living agents. Now, animals that carry pathogenic microorganisms are called vectors. Remember, I said that carriers of microorganisms are agents that pick microorganisms from one place to another. Now, these carriers can be animals, that is living, non-living. They are air, water, and food. Now, but the animals that carry pathogenic, meaning harmful microorganisms, are called vectors. Please don't forget that. Vectors are animals that carry pathogenic microorganisms. That's the definition of vectors. Please put that at the tip of your fingers. Vectors are animals that carry pathogenic microorganisms. Now, let's look at some vectors. Please listen. I want you to listen here now. The examples are, please, you will rule a table with 3 columns. Rule a table. I could not make this a table. Please listen. For those that Blaze and Co, you're just writing. I want you to listen. Under these examples, you rule a line. 3 columns is a table. The first will have vectors. Second will have the bacteria. And then the third will have the name of the disease. I could not do that. That is why you see a dash. Let me show you. Number 1, you have female Anopheles mosquito. That is the vector. Now, what does the female Anopheles mosquito carry? That's the parasite, is Plasmodium. So your 3 table will be vector, name of vector, the causative organism, then the diseases they spread. So under the first one, you have female Anopheles mosquito. Then you can see there's a dash. Then you write Plasmodium is the causative organism. That's what this female Anopheles mosquito can pick. Then malaria is the disease caused by the female Anopheles mosquito. Then you go back to the next one, Sesefly. Sesefly is the vector. Trypanosome is the name of the causative organism that is picked by Sesefly. Now, the disease caused is called trypanosomiasis or sleeping sickness. That is why you see there's a dash here and a dash there. So rule your line so that you understand the table very well. You have Sesefly, trypanosome, then disease caused by trypanosome or Sesefly is sleeping sickness, which is also known as trypanosomiasis. Then the third is housefly. Housefly is the vector. Then what is the causative organism or what parasite? Is Vibrio cholera. The disease that is caused by housefly or that parasite, causative organism, which is Vibrio cholera, is cholera and typhoid fever. Cholera and typhoid fever. Daniel Eze, are you hearing me? Daniel Eze, are you hearing me? Okay. Yes, so please rule your line, make a table in your notes. You write female Anopheles mosquito on the first column. The second will be Plasmodium. Agungu, are you following me? Agungu. And then you have, please, sleeping sickness is also called trypanosomiasis. So you can write that in full. Now, the next one, number 4 and 5. Number 4 and 5, you have rat flea or body louse. The causative organism is Rickettsia, and it causes typhus. Then you have the next one is Aedes. Aedes mosquito can pick virus, and then it causes yellow fever or dengue fever. Then number 6 is rat fleas. The causative organism is bacterium, and the disease that is spread is plague. Please complete it. I gave you 6. You can also add as much as you can get. Rat fleas causative organism, the virus or what it picks, the infection is rickettsia, and then it causes disease called typhus. Aedes mosquito pick virus and can cause yellow fever or dengue fever. Rat fleas will pick bacterium and then will cause plague. Is that okay? Please nod your head. Okay, correct.
Yes,
I like that response. Now let's move to the next one. Growth of microorganisms. Growth of microorganisms. Microorganisms do increase in sizes because they are living things and they multiply in number of cells using either the culture medium provided or any suitable surface. They can use the culture medium, that's the agar you provide for them. They grow in it, they increase in size and multiply. Or any suitable surface, even your skin, microorganisms can grow and multiply. So they grow because they are living things, because living things grow is one of the major characteristics. Now let's look at measurement of the growth, or measuring growth of microorganism. How do you measure the growth of microorganism? Yes,
Ma'am, please go back to the former slide.
Okay, this is the former slide. Or is it the vectors? Okay.
Yes.
This is the beginning of the vectors. I said animals that carry pathogenic microorganisms are called vectors. Example, I've told you what to do. Female Anopheles mosquito, you rule a table, a 3-column, Plasmodium malaria fever. Then fly, it picks trypanosome, that's the causative organism for sleeping sickness, which is also called trypanosomiasis. And then you have housefly, vibrio cholera, cause disease that is caused by vibrio cholera is cholera and typhoid. Then you have the next one, rats and body louse, rickettsia, typhus. Aedes mosquito will pick virus and will cause yellow fever. The same as rat fleas will pick bacteria and then will cause plague. Now we started discussing growth of microorganisms. I said microorganisms do increase in sizes because they are living things and they multiply in the number of cells using either the culture medium provided or any suitable surface. Now we want to look at measuring growth of microorganisms. Measuring growth of microorganisms. We have two methods of measuring growth, but I picked the first one. Now, the first method, a sample of a clear liquid culture medium is inoculated into a nutrient agar or agar. A sample of a clear liquid, it could be water, is inoculated, means is introduced into a nutrient agar. Now, as the bacteria increases, it becomes cloudy and turbid. As the bacteria increases, it becomes what? Cloudy and turbid. Now, progressive increase in turbidity indicates an increase in the number of bacterial cells. Now, there's an instrument we use to measure it when they have increased and become cloudy. There's an instrument you use. It is called spectrophotometer. It's used to measure the turbidity in the laboratory of that nutrient agar. Now, thus, as you measure the turbidity at interval, you can also use that to ascertain the growth of the bacteria. I said spectrophotometer is used to measure the turbidity in the laboratory. Thus, it enables you to ascertain or determine the growth of microorganisms in the lab. Now let's continue quickly. Now I want to look at beneficial effect of microorganisms. Now, remember I said last week that some microorganisms are harmful, some are harmless. Now, these harmless microorganisms can be beneficial. Some bacteria and fungi are beneficial to man in nature, in medicine, and in industries, in the industries. So we have three areas in which microorganisms are beneficial to man. Now some of these bacteria and fungi, they are beneficial to us in nature, some in medicine, and some in the industries. Now let's look at in nature, how do microorganisms, either bacteria or fungi, how do they benefit man in nature? Now please listen. In nature, I just wrote the points, number 1, number 2, like that. You would have to explain. As I'm explaining, please indicate. Number 1, you put a semicolon. Are you listening to me?
Yes, ma'am.
Because most of you, you're just busy writing. Please listen and understand what I want you to do. Now, in nature, number 1 is compost formation. You put a semicolon like this. Look at your screen. Put a semicolon like this and explain. Say something. Explain. You can also write what I'm saying. That is why you ought to listen. You have to listen. Compost formation. Some bacteria are used for, compost means they, like fertilizers. They increase soil fertility. So they are decomposers, that's saprophytes. I said that before. So especially fungi. That some fungi are saprophytes. So compost formation means as they decompose dead plant and animal in the soil, as they do that, they form compost, or even your dung, your refuse, where you pour your refuse. As fungi decomposes those refuse, it acts as compost to the soil. The second is nitrogen fixation. The second is nitrogen fixation. Some bacteria, like if you remember, we've talked about nitrogen. When we talked about nitrogen cycle, I said Rhizobium leguminosaureum is a bacteria that fixes nitrogen in the root nodules of most leguminous plants.
So that you can use for the Zoom.
So nitrogen fixation is another benefit of bacteria in nature, because some bacteria fixes nitrogen in plant. Example is Rhizobium leguminosaureum. Now let's move to the next one, maintaining soil fertility. I've talked about that just now. Number 3, most fungi, as they decompose refuse and other organisms in the soil, they do that to form compost. Compost means manure, and thereby increasing soil fertility. Now, number 4 here is digestion of cellulose. So many bacteria are in the gut of ruminants. Gut means alimentary canal of ruminants. Animals that are ruminants. So they help to digest cellulose. Decomposition means saprophytes. Fungi are saprophytes. To decompose means they feed on, they break down dead plants and animals. That is what we mean by decomposition. So fungi decompose, and we have silage making, sewage treatment. most bacteria are used to treat our sewage, break them down, treat them, especially in the socker way. So that's that for beneficial effect of microorganisms in nature. Now, in medicine, what are the beneficial effects? One, they are used for manufacturing drugs like penicillin. Microorganisms are used to manufacture drugs. They are also used as antibiotics. So source of vitamin B, some bacteria are used as source of vitamin B, and then sources of enzymes. So in medicine, the beneficial effects of microorganisms include manufacturing of drugs and vaccine, sources of vitamin B, and then sources of enzymes. So you would explain this. Then in the industries, microorganisms are used for baking, like your yeast. Your yeast is your fungi. It is used for baking. When you apply a little quantity of yeast in bread, it will make the dough to rise. So preparation of alcoholic drink, most alcoholic drink contains bacteria. Making of cheese and yogurt, that's the harmful, not the pathogenic microorganisms now, the harmless microorganisms. They are used to make cheese and the yogurt you drink. That's for industries. And then we have rating jute, curing of tobacco, etc. These are some of the beneficial effects of microorganisms in the industries. Now, what about the harmful effect? What are the harmful effects of microorganisms? They cause diseases. They cause diseases, spoilage of food. Most microorganisms can damage our food. So spoilage of food. They cause death. Most microorganisms cause death. Typhoid fever. If a patient is having untreated typhoid for a very long time, it can lead to death. And then it causes undesirable quality of product, and then reduction in yield for farmers. So that's that. This is harmful effect of microorganism, causes of disease. It causes diseases, spoilage of food, and then it causes death. So this is the last part, undesirable quality of product and reduction in yield. Then the next one is definition of some terms. That is under this topic, diseases. What are diseases? A disease is a condition in which the normal functions of an organism or part of the body or cell or organ is impaired or disturbed. When part of your organ or body is impaired or disturbed, we say that person has contacted or is infected with a disease or the other. Now, health. Health is a state of alertness or freedom from ill health. When you are not sick, you are in health. That's a state of alertness. It's a state of alertness. A state of alertness and freedom from ill health. That is what we mean by health. Now, number 3, you have symptoms. What are symptoms? Observable effects. You know, I said effect can be positive or negative. So symptoms are observable effects on an organism caused by the presence of a disease or pathogen. Pathogen means harmful microorganisms. So we've talked about disease, health, and then symptoms. Now, other terms that are used under this topic is pathogens. You know what pathogens are? Pathogens are harmful microorganisms. So you can write that, please. Others are pathogens. Please explain or define pathogens. Vector, I just finished explaining vector. that a vector is an animal that carries pathogenic microorganism, then antibody, then vaccination. So what is vaccination? Vaccination is the introduction of a mild pathogen into the body. Mild pathogen means it is not as harmful as what maybe a patient is suffering from. Now, let me explain this. Please listen. When you go for a test, you want to test your blood sample for typhoid, if there's a typhoid, because if there's typhoid in the blood, or malaria. Now, what happens is, as they grow or as they culture your sample, the blood sample, they will bring other microorganisms. Now, like penicillin, like Cipro, they will bring some of this. Now, if these mild pathogens are resistant to it, that becomes a cure. That is what we call vaccination. Vaccination is the introduction of a mild pathogen into the body so as to develop resistance or immunity against that pathogen as the animal growth. Blaze, you raised up your hand.
No, ma, it was a mistake.
Okay, my dear, let's continue. So let's go back. I was explaining vaccination. I said other terms we use is pathogen. Who is that singing? We have pathogens, we have vector, antibody, and vaccination. I've explained pathogen, so please include that. Write it as your number 4, number 5, vector, number 6, antibody, and then number 7, vaccination. Now, what is vaccination? I said is the introduction of a mild pathogen into the body so as to develop resistance or immunity against that pathogen as the animal grows. Now, let's move forward. What are those diseases caused by viruses? I gave these are examples: poliomyelitis, infective hepatitis, measles, common cold, smallpox, chickenpox. Because sometimes in your WAEC, you might be asked, name 5 diseases caused by viruses, so that you won't write typhoid. Typhoid is not caused by a virus. Please take note. Why I'm doing this is for you to know. These are some of the diseases you might mistake these diseases for a bacterial disease or fungi. Yellow fever is a disease caused by virus. Rabies, AIDS, acquired immunodeficiency syndrome, AIDS. And then you have common cold, measles. Measles is caused by viruses, smallpox, etc. So these are some animal diseases caused by viruses. Now let's look at other animal diseases caused by bacteria and fungi. Our time is fast spent. Animal diseases caused by bacteria, you have tuberculosis, leprosy, tetanus. You know tetanus, when a rusted nail puncture any part of your body, it can lead to tetanus if it's exposed. That's bacteria. That is why quickly some of you you go to take an injection to kill that. Now you also have typhoid. Typhoid is caused by bacteria. Dysentery, gonorrhea, syphilis, cholera, whooping cough. These are diseases, animal diseases. Please take note, they are not plant diseases, because in your WAEC, please listen, in your WAEC, you might be asked this. There are some plant diseases caused by viruses, bacteria, and fungi. Is different from animal diseases. Please don't mistake that. Animal diseases caused by viruses, they are this. Now you also have plant diseases caused by viruses, like cassava mosaic and the rest. You are going to write that as part of your assignment. So you have This animal disease is caused by bacteria, tuberculosis, leprosy, tetanus, typhoid, dysentery, gonorrhea, syphilis, cholera, whooping cough. Now, animal disease is caused by fungi, ringworm, aspergillosis, athletic foot. So these are some of the diseases. So now this is what you will do for me. Number one, you discuss, this is your assignment, you discuss an experiment on how to identify microorganisms. That is one. Number two, you will list 5 diseases each caused by viruses, fungi, and bacteria in plants. In plants, not animal diseases now, but plant diseases caused by viruses, fungi, and bacteria. Any question?
Ma,
Yes.
what is red?
Huh?
What is writing juice?
It's a kind of when you sap, when you get, let me go back to it. I think it's one of the beneficial effects of microorganism, isn't it? It's in
Yes,
your
ma'am.
textbook, look at it, written jute. You kind of get it, it's used to make fiber. You will see most of the terms in your textbook is used to make fiber. Written jute is used to make fiber. Curing of tobacco too is to get to tobacco. People that uses tobacco. Is that okay?
Yes, ma'am.
Okay, what else? Any question? Let me go back to your assignment. Please write down your assignment. I said discuss an experiment on how to identify microorganisms, then list 5 diseases of plants. That's diseases each caused by viruses, fungi, and bacteria in plants, just as I gave you this for animal. You do that of plants. Any other question?
Ma'am, please can you send the notes?
Yes, I will. In a jiffy. I will do that after the class. Any other questions so we can pray? Father, we thank you because we have come to the end of our class.
Amen.
We ask that you protect and help us in Jesus' name.
Amen.
Okay. All right. I'll see you next week. Please do your assignment and complete it.
Okay.
Okay, ma. Bye bye, ma.
All right.